Secondary growth. The growth of the roots and stem in length with the help of apical meristem is called the primary growth. Apart from primary growth, most dicotyledonous plants exhibit an increase in girth. This increase is called the secondary growth. The tissues involved in secondary growth are the two lateral meristems, that is vascular cambium and cork cambium. Vascular cambium. The meristematic layer that is responsible for cutting off vascular tissues, xylem and phloem, is called vascular cambium. In the young stem, it is present in patches as a single layer between the xylem and phloem. Later, it forms a complete ring. Formation of cambial ring. In dicot stems, the cells of cambium present between primary xylem and primary phloem is called intrafascicular cambium. The cells of medullary rays adjoining these intravascular cambium becomes meristematic and form the interfascicular cambium. Thus, a continuous ring of cambium is formed. Activity of the cambial ring. The cambial ring becomes active and begins to cut off new cells both towards the inner and the outer sides. The cells cut off towards pith mature into secondary xylem, and the cells cut off towards periphery mature into secondary phloem. The cambium is generally more active on the inner side than on the outer. As a result, the amount of secondary xylem produced is more than secondary phloem and soon forms a compact mass. The primary and secondary phloems get gradually crushed due to continued formation of and accumulation of secondary xylem. The primary xylem, however, remains more or less intact or in or around the center. At some places, the cambium forms a narrow band of parenchyma which passes through the secondary xylem and the secondary phloem in the radial directions. These are the secondary medullary rays. Figure 6.9. Secondary growth in a dicot stem. Diagrammatic. Stages in transverse views. Spring wood and autumn wood. The activity of cambium is under control of many physiological and environmental factors. In temperate regions, the climatic conditions are not uniform throughout the year. In the spring season, cambium is very active and produces a large number of xylary elements having vessels with wider cavities. The wood formed during this season is called spring wood or early wood. In winter, the cambium is less active and forms fewer xylary elements that have narrow vessels, and this wood is called autumn wood or late wood. The spring wood is lighter in color and has lower density, whereas the autumn wood is darker and has higher density. Two kinds of woods that appear as alternate concentric rings constitute an annual ring. Annual rings seen in a cut stem give an estimate of the age of the tree. Heartwood and sapwood. In old trees, the greater part of secondary xylem is dark brown due to deposition of organic compounds like tannins, resins, oils, gums, aromatic substances, and essential oils in the central or innermost layer of the stem. These substances make it hard, durable, and resistant to the attacks of microorganisms and insects. This region comprises dead elements with highly lignified walls and is called heartwood. The heartwood does not conduct water, but it gives mechanical support to the stem. The peripheral region of the secondary xylem is lighter in color and is known as sapwood. It is involved in conduction of water and minerals from root to leaf. Cork cambium. As the stem continues to increase in girth due to the activity of vascular cambium, the outer cortical and epidermal layers get broken and need to be replaced to provide new protective cell layers. Hence, sooner or later, another meristematic tissue called cork cambium or phellogen develops, usually in the cortex region. Phellogen is a couple of layers thick. It is made up of narrow, thin-walled and nearly rectangular cells. Phellogen cuts off cells on both the sides. The outer cell differentiate into cork or phellum, while the inner cells differentiate into secondary cortex or phelloderm. The cork is impervasive to water due to suberin deposition in the cell wall. The cells of secondary cortex are parenchymatous. Phellogen, phellum and phelloderm are collectively known as periderm. Due to the activity of cork cambium, pressure builds up on the remaining layers peripheral to phellogen, and ultimately these layers die and slug off. Bark is a non-technical term that refers to all tissues exterior to the vascular cambium, therefore including secondary phloem. Bark refers to a number of tissue types, namely periderm and secondary phloem. Bark that is formed early in the season is called early or soft bark. Towards the end of the season, late or hard bark is formed. Name the various kinds of cell layers which constitute the bark. At certain regions, the phylogen cuts off closely arranged parenchymatous cells on the outer side instead of cork cells. These parenchymatous cells soon rupture the epidermis, forming a lens-shaped opening called lenticels. Lenticels permit the exchange of gases between outer atmosphere and the internal tissue of the stem. These occur in mostly woody trees. Secondary growth in roots. In the dicot root, the vascular cambium is completely secondary in origin. It originates from the tissue located just below the phloem bundles. A portion of pericycle tissue above the protoxylem formed a complete and continuous wavy ring, which later becomes circular. Further events are similar to those already described above for a dicotyledonous stem. Figure 6.10. A. Lenticel and B. Bark. Figure 6.11. Different stages of secondary growth in a typical dicot root. Secondary growth also occurs in stems and roots of gymnosperms. However, secondary growth does not occur in monocotyledons.